Welcome to PG Pathshala Biophysics chapter. Today we'll be talking about the module 4, which is about the regulators and checkpoints of cell cycle. And I'm Dr. Ruby Dhar and I'm a Biocure Fellow DBT. So we have discussed in module 3 about how cells grow and then divide following the various phases of cell cycle, which consist of multiple steps and needs a fine-tuned coordination among themselves. In order to make sure everything goes right, there are checkpoints in the cell cycle. Now, lack of proper regulation at the checkpoints leads to uncontrolled cell division or failure to proceed into the cell division, finally leading to cancer. Now, just to give a brief introduction about the chapter, the cell cycle proceeds by a defined sequence of events where late events depend upon completion of early events. The aim of the dependency of the events is to distribute complete and accurate replicas of the genome to daughter cells. To monitor this dependency, cells are equipped with the checkpoints that are set at various stages of the cell cycle. When cells have DNA damage that have to be repaired, cells activate DNA damage checkpoint that arrests the cell cycle. According to the cell cycle stages, DNA damage checkpoints are classified into three main categories. One is the G1 to S checkpoint, an intra-S phase checkpoint, and G2M checkpoint. Upon perturbation of DNA replication by drugs that interfere with DNA synthesis, DNA lesions, or obstacles on DNA, cells activate DNA replication checkpoint that arrests the cell cycle at G2M transition until DNA replication is complete. There are more checkpoints such as spindle checkpoint and morphogenesis checkpoint. The spindle checkpoint arrests the cell cycle at M phase until all the chromosomes are aligned on the spindle. Now, this checkpoint is very important for equal distribution of chromosomes. Morphogenesis checkpoint detects abnormality in cytoskeleton and arrests cell cycle at G2M transition. Beside these internal checkpoints, cell cycle is regulated by various external and internal factors, which we are going to discuss in this chapter in further details. So, this chapter has the following objectives. So, we will begin with eukaryotic cell cycle and its duration, the length of cell cycle, frequency of cell division in different cell types, regulation of the cell cycle by external events, regulation of the cell cycle by the internal checkpoints, and the various checkpoints as we discuss in the introduction, and then the regulator molecules of the cell cycle. This can be the internal regulators, the external regulators. So, and we will be talking about various cyclins, the ubiquitin ligase, and the various kinase and the phosphatases which are involved in cell cycle. And finally, as we know that dysregulation in cell cycle leads to cancer, and we'll talk about the oncogenes and the tumor suppressor genes. Now, starting with the first objective, eukaryotic cell cycle and its duration. So, eukaryotic cell cycle of most cells consists of 4 coordinated process during the, so this is like cell growth, the DNA replication, distribution of the duplicated chromosome to the daughter cells, and finally the division of the cell into 2 daughter cells. The cell cycle in eukaryotes is a very complex and consists of 4 discrete phases. Although cell growth is usually a continuous process, DNA is synthesized during only one phase of the cell cycle, and the replicated chromosomes are then distributed to daughter nuclei by a complex series of events preceding cell division. Progression between these stages of the cell cycle is controlled by a conserved regulatory apparatus which not only coordinates the different events of the cell cycle, but also links the cell cycle with extracellular signals that control cell proliferation. Now, in bacteria, cell growth and DNA replication take place throughout most of the cell cycle, and duplicated chromosomes are distributed to daughter cells in association with the plasma membrane. Now, length of the cell cycle. So, as I just discussed in the introduction, that various cells have different length. Now, the length of the cell cycle is highly variable even within the cells of a single organisms. Like in humans, the frequency of cell turnover ranges from a few hours in early embryonic development to an average of 2 to 5 days for epithelial cells, and to an entire human lifetime spent in D0 by specialized cells such as the cortical neurons or the cardiac muscle cells. The time that a cell spends in each phase of the cell cycle also varies in each phase. When fast dividing mammalian cells are grown in culture, that is in vitro when we are maintaining them under proper growth conditions, the length of the cell cycle is about 24 hours. In rapidly dividing human cells with a 24-hour cell cycle, the different phases undergo approximately the following distribution. So, the G1 phase lasts approximately 9 hours, S phase lasts 10 hours, and the G2 phase lasts about 4 and 1.5 hours, and the M phase lasts approximately, it's the shortest phase, and it lasts just within 30 minutes. In early embryos of fruit flies, the cell cycle is completed in about 8 minutes. So, we can see that how variation there is between the various length of the cell cycle. The timing of events in the cell cycle is controlled by mechanisms that are both internal and external to the cell. So, next, regulation of the cell cycle by the external events. Both the initiation and inhibition of cell division are triggered by events external to the cell when it is about to begin the replication process. Whatever the source of the message, the cell receives the signal, and a series of events within the cell allows it to proceed into interphase. Moving forward from this initiation point, every parameter required during each cell cycle phase must be met or the cycle cannot progress. The following external factors can affect the progress into the cell cycle. Now, death of a nearby cell. So, if the cell is surrounded by a lot of dead cells, and the dead cells release a lot of toxin and other factors which can inhibit the growth of the neighboring cell, presence of growth hormone and cytokines. Crowding of cells can also inhibit cell division because that will lead to the scarcity of the nutrients. Cell size, as the cell grows, it becomes inefficient due to its decreasing surface to volume ratio. The solution to this problem is to undergo cell division and initiate cell cycle. Now, regulation at internal checkpoints. It is essential that the daughter cells produced be exact duplicates of the parent cell. Mistakes in the duplication or distribution of the chromosomes lead to mutations that may be passed forward to every new cell produced from an abnormal cell. Now, there are internal control mechanisms that operate at 3 main cell cycle checkpoints to control the cell cycle. A checkpoint is one of several points in the eukaryotic cell cycle at which the progression of a cell to the next stage in the cycle can be halted until conditions are favorable. There are 3 important checkpoints, and they occur near the end of G1, at the G2M transition, and during metaphase. Now, we will begin with the discussion about the G1 checkpoint. The G1 checkpoint determines whether all conditions are favorable for cell division to proceed. The G1 checkpoint, also called the restriction point, in yeast, where it was first discovered, is a point at which the cell irreversibly commits to the cell division process. External influences such as growth factors play a large role in carrying the cell past the G1 checkpoint. Now, various factors determine the cell's fate to pass through the G1 phase. Now, the external influences like growth factors, adequate reserves and cell size, damage in genomic DNA, and then the cell can halt the cycle and try to rectify it. So, once it is restricted at a particular checkpoint, it will pause and try to see if it can repair, or it can advance into G0 and await further signals when conditions improve. Now, next is the G2 checkpoint. The G2 checkpoint bars entry into the mitotic phase if certain conditions are not met. At this checkpoint, similar to the G1 phase, cell size and protein reserves are assessed. Ensure all chromosomes have been replicated and that the replicated DNA is not damaged. If the checkpoint mechanisms detect problems with the DNA, the cell cycle is halted and the cell attempts to either complete DNA replication or repair the damaged DNA. The other important checkpoint is the M checkpoint, the M phase checkpoint. The M phase checkpoint occurs near the end of the metaphase stage of karyokinases. The M checkpoint is also known as the spindle checkpoint because it determines whether all the sister chromatids are correctly attached to the spindle microtubules. Kinetochores assemble at centromeres and attach the chromosome to the mitotic spindle fibers. So, if you see the figure, there is a good elaboration about all these stages, and in metaphase, chromosomes line up on the metaphase plate in the center of the spindle. Separation of the sister chromatids during anaphase is an irreversible step. The cycle will not proceed until the kinetochores of each pair of sister chromatids are firmly anchored to at least two spindle fibers arising from opposite poles of the cell. Now, we are next moving to the regulator molecules of the cell cycle. So far we discussed about the internal checkpoints. Now, what are the other molecules which regulate the cell cycle? So, these regulatory molecules either promote progress of the cell to the next phase, this is called the positive regulation, or halt the cycle, which is called negative regulation. Now, regulator molecules may act individually or they can influence the activity or promotion of other regulatory proteins. Therefore, the failure of a single regulator may have almost no effect on the cell cycle, especially if more than one mechanism controls the same event. So, depending on the requirement or the conditions of that particular cell cycle, it is decided that removing a growth factor or hormone is going to affect the cell cycle or not. Now, there are three main groups of intracellular molecules that control the various checkpoints of the cell. So, inside the cell there are different groups of proteins. The three important ones we are going to discuss in this module, and these are number 1, the cyclins and the cyclin dependent kinases. Number 2, the ubiquitin protein ligases. Number 3, the protein phosphatases family. So, starting off with the cyclins and cyclin dependent kinases, two groups of proteins called cyclins and cyclin dependent kinases, or the CDKs, are responsible for the progress of the cell through the various checkpoints and decides the entry of non-dividing cells into the cell cycle. The mechanism regulating the progression of cells through their division cycle is highly conserved in evolution, and plants have retained the basic components of this mechanism. The various activities of the cell cycle then are determined by the combination of cyclins and the CDKs that are active at each stage as shown in the table. So, the level of the 4 cyclin proteins fluctuate throughout the cell cycle in a predictable pattern, as we can see. So, the G1 cyclins coordinate with CDKs 4 and 6. So, here cyclin D and the CDK 4 and 6 play a role and can react to outside signals such as growth factors or mitogens. Now, in the G1 to S cyclins, cyclin E and A play a role and they interact with CDK2 and regulate centrosome duplication. In S cyclins, cyclin E, A coordinate with again CDK2 and targets are the helicases and the polymerases. And in the mitosis cyclins, cyclin B play a role and coordinate with CDK1 and it regulates the G2M checkpoint. So, as we see, there is a distribution of fixed cyclins and CDKs. Now, increase in the concentration of cyclin proteins is triggered by both external and internal signals. After the cell moves to the next stage of the cell cycle, the cyclins that were active in the previous stage are degraded. Cyclins regulate the cell cycle only when they are tightly bound to CDKs. So, there is a strong coordination between these two. To be fully active, the CDK and cyclin complex must also be phosphorylated in specific locations. Like all kinases, CDKs are enzymes. These are kinases that phosphorylate other proteins. Phosphorylation activates the protein by changing its shape. The proteins phosphorylated by CDKs are involved in advancing the cell to the next phase. Now, the levels of CDK proteins are relatively stable throughout the cell cycle. However, the concentration of cyclin fluctuate and hence they got the name cyclin because they keep changing during every cycle of this phase. CDK cyclin complex, the different cyclins and CDKs bind at specific points in the cell cycle and thus regulate different checkpoints, and as we have shown in the table, they form specific complex during a particular stage of cell cycle. Next is the regulation by the ubiquitins. The proteolysis of many core components of the cell cycle machinery is controlled by two major classes of ubiquitin ligases. The SCF, which stands for SKP1 Cul1 F-box ROCK and RBX complexes, and another is the APC, which is the anaphase promoting complex or the cyclosome. Now, beginning with the SCF complex. In humans, 68 SCF ligases have been identified to date, each characterized by a different F-box protein subunit that provides specificity by directly recruiting the substrate to the rest of the ligase and ultimately to the ubiquitin conjugating enzyme. Host SCF substrates are recognized and bound by the F-box protein subunit only when they are phosphorylated on specific sites. Notably, only 3 out of 68 human SCF ubiquitin ligases have well-established substrates, many of which are involved in cell cycle control. And then next is the APC complex. The APC ligase exists in two forms. First is activated by CDH1, which helps to keep CDK activity low and therefore to maintain the G0 G1 state. Another route is activated by the CDC20, which is necessary for progression through mitosis and for inactivation of CDK1, thereby allowing the exit from mitosis. Now, the APC appears to have taken on a number of additional tasks both within and outside of the cell cycle, including the degradation of the mitotic kinases such as polo-like kinases and the NEMA-related kinases and the Aurora kinases, proteins involved in spindle function, for example, kinesins like KIP1, SYN8 and XKID, or proteins that function in DNA replication such as CDC6, DBF4, geminin. Notably, SCF ligases and APC reciprocally control each other. In G1, APC CDH1 promotes the degradation of SKP2, thereby maintaining the G1 state, whereas in early mitosis, SCF phosphorylation activates APC CDC20 by inducing the degradation of its inhibitor EMI1. Now coming up to the third internal regulator, which is the protein phosphatases family. The first major group is the protein tyrosine phosphatase PTP family, which includes the dual specificity tyrosine and serine threonine phosphatases like DUSPs. We call them dual phosphatases. Now, considering that CDK1 has hundreds of substrates and that several other protein kinases such as Aurora A, B, Polo, V1 and MIT1 are involved in entry into mitosis and mitotic progression, several different protein phosphatases are probably involved in the reversal or regulation of these processes. V1 is the main kinase that phosphorylates the tyrosine 15 residue of CDK1, the dephosphorylation of which by CDC25 phosphatase is essential for the activation of CDK1. CDK1 mediated phosphorylation activates CDC25, whereas it turns off V1. Thus, CDC25 and V1 form positive and negative feedback loops respectively with CDK1. Second main group of phosphatases, the serine threonine specific phosphatases, we call them PSTPs, and there are many roles in mitosis and cytokinesis. PSTPs can be further subdivided into ppm family of metallodependent phosphatases including ppm1 and the phosphoprotein phosphatases PPP family, which are both dependent on metal ions for catalysis. Now, the ppm family members function in signal transduction and DNA damage pathways, and it is therefore the PPP family that is most commonly associated with mitotic regulation. Remarkably, brief treatment of cells with the algal toxin ocadaic acid, which is specific for the PPP enzyme, trigger many of the changes that are associated with mitosis, including chromatin condensation and structural rearrangement of organelles such as the Golgi apparatus. This finding indicates that mitotic entry is normally opposed by PPP enzymes and suggests that inhibition of the PPP activity and not simply protein kinase activation is required to allow cells to progress into mitosis. Now, the major topic which affects the cell cycle is leading to cancer. So, cancer and cell cycle. Cancer comprises many different diseases caused by common mechanisms, uncontrolled cell growth. Despite redundancy and overlapping levels of cell cycle control, errors do occur. One of the critical processes monitored by the cell cycle checkpoint surveillance mechanism is the proper replication of DNA during the S phase. Even when all the cell cycle controls are fully functional, a small percentage of replication error, that means a mutation, will be passed on to the daughter cells. If changes of the DNA nucleotide sequence occur within a coding portion of a gene and are not corrected, a gene mutation results. All cancers start when a gene mutation gives rise to a faulty protein that plays a role in cell reproduction. The change in the cell that results from the malformed protein may be minor, perhaps a slight delay in the binding of CDK to cyclin or an RB protein that detaches from its target DNA while still phosphorylated. Even minor mistakes, however, may allow subsequent mistakes to occur. So, it's like a relay race. Over and over, small uncorrected errors are passed from the parent cell to the daughter cells and amplified as each generation produces more non-functional proteins from uncorrected DNA damage. Eventually, the pace of the cell cycle speeds up as the effectiveness of the control and repair mechanism decreases. Uncontrolled growth of the mutated cells outpaces the growth of normal cells in the area, and a tumor can result. Now, the two main categories, the genes which affect the cell cycle growth, are proto-oncogenes and suppressor genes. So, genes which promote cell cycle growth are called the proto-oncogenes, and these are normal genes that when mutated in certain ways become oncogenes, genes that cause a cell to become cancerous. So, it is promoting cell proliferation and thus cell growth. So, in most instances, the alteration of the DNA sequence will result in a less functional or non-functional protein. The result is detrimental to the cell and will likely prevent the cell from completing the cell cycle. However, the organism is not harmed because the mutation will not be carried forward. If a cell cannot reproduce, the mutation is not propagated and the damage is minimal. Occasionally, however, a gene mutation causes a change that increases the activity of a positive regulator. For example, a mutation that allows CDK to be activated without being partnered with cyclin and could push the cell cycle past a checkpoint before all of the required conditions are met. So, it is not restricted now or not being checked in any restriction point. So, the resulting daughter cells are too damaged to undergo further cell division. The mutation would not be propagated and no harm would come to the organism. However, if the atypical daughter cells are able to undergo further cell division, subsequent generation of cells will probably accumulate even more mutations, some possibly in additional genes that regulate the cell cycle. The CDK gene in the above example is only one of many genes that are considered as proto-oncogenes. In addition to the cell cycle regulatory proteins, any protein that influences the cycle can be altered in such a way as to override the cell cycle checkpoints are categorized as oncogenes. An oncogene is any gene that when altered leads to an increase in the rate of cell cycle progression. So, the other examples of oncogenes are the EGFR, the receptor of the epidermal growth factor that is involved in growth factor mediated signaling, a very well known example in breast cancer and prostate cancer, the KDR, the receptor for VEGF, vascular endothelial growth factor that is involved in angiogenesis, and SRC kinases. So, there are various examples. Now, we are switching gears to the tumor suppressors. So, tumor suppressors are the ones which negatively regulate the cell cycle. Tumor suppressor genes are segments of DNA that code for negative regulator proteins, the type of regulators that when activated can prevent the cell from undergoing uncontrolled division. The three classical best understood tumor suppressor proteins are RB, P53 and P21, and their function is to put a roadblock to cell cycle progression until certain events are completed. A cell that carries a mutated form of a negative regulator might not be able to halt the cell cycle if there is a problem. Tumor suppressors are similar to brakes in a vehicle. Malfunctioning brakes can contribute to a car crash. So, here malfunctioning tumor suppressor will lead to more cell cycle progression and growth. So, mutated P53 has been isolated in many human tumor cells. The discovery is not surprising. in light of the multiple role of p53 protein plays in the regulation of the G1 checkpoint. A cell with a faulty p53 may fail to detect any mutation in genetic DNA. It may no longer able to signal the necessary DNA repair enzyme. So, even if there is a mutation, it is not able to repair. Either way, undamaged DNA will remain uncorrected. At this point, a functional p53 may deem the cell unsalvageable and trigger programmed cell death, which is called apoptosis. The damaged version of p53 found in cancer cells, however, cannot trigger apoptosis. The loss of p53 function has other repercussions for the cell cycle. Mutated p53 might lose its ability to trigger p21 production. Without adequate levels of p21, there is no effective block on CDK activation. Essentially, without a fully functional p53, the G1 checkpoint is severely compromised and the cell proceeds directly from G1 to S regardless of internal and external conditions. At the completion of this shortened cell cycle, two daughter cells are produced that have inherited the mutated p53 gene. Given the non-optimal conditions under which the parent cell reproduced, it is likely that the daughter cells will have acquired other mutations in addition to the faulty tumor suppressor genes. Cells such as these daughter cells quickly accumulate both oncogenes and non-functional tumor suppressor genes, thus leading to tumor growth. Now, another interesting tumor suppressor is the RB, and it exerts its regulatory influence on other positive regulator proteins. Chiefly, RB monitors cell size. In the active dephosphorylated state, RB binds to proteins called transcription factors, most commonly the E2F. Transcription factors turn on specific genes, allowing the production of proteins encoded by that gene. When RB is bound to E2F, production of proteins necessary for G1S transition is blocked. As the cell increases in size, RB is slowly phosphorylated until it becomes inactivated. Now, RB releases E2F, which can now turn on the gene that produces the transition protein, and this particular block is removed. For the cell to move past each of the checkpoints, all positive regulators must be turned on and all negative regulators must be turned off. Now, to summarize this module, as we have seen, the cell cycle in eukaryotes is a very complex and consists of 4 discrete phases. Although cell growth is usually a continuous process, DNA is synthesized during only one phase of the cell cycle, and the replicated chromosomes are then distributed to daughter nuclei by a complex series of events preceding cell division. Now, another point which we discussed is the length of the cell cycle is highly variable even within the cells of a single organism. There are external and internal regulators of cell cycle. External factors include various growth factors, neighboring cells, health condition, secretion by the neighboring cells, cell's own size. Now, fifth point is the internal factors include the 3 important checkpoints G1, G2 and M. The G1 checkpoint determines whether all conditions are favorable for cell division to proceed. The G2 checkpoint bars entry into the mitotic phase if certain conditions are not met. The M checkpoint is also known as the spindle checkpoint because it determines whether all the sister chromatids are correctly attached to the spindle microtubules. In addition to these internal checkpoints, there are 3 groups of intracellular molecules that regulate the cell cycle, and these are the cyclins family, the coordination between the cyclin and the CDKs, ubiquitin ligases and the protein phosphatases. The genes that code for the positive cell cycle regulators are called proto-oncogenes, and examples as we discussed are EGFR, VEGF and SRC, to name few of them. Unlike the proto-oncogenes, there are a group of genes which negatively regulate the cell cycle. These are known as the tumor suppressor genes. The three classical best understood tumor suppressor genes are the retinoblastoma RB, P53 and P21. Their role is to put a roadblock to the cell cycle progression until certain events are completed and the cell is ready to move into the cell cycle. So, with this I complete this module and thank you very much for your attention.